I really enjoy population genetics because it's a way of seeing how the genotypes in populations shift over time in response to external forces like natural selection or even human selection. Now, what I want to do is work through the example that I have in the text because sometimes it's hard to follow when it's just words on a page. So I'm going to step through it with you. The scenario is that I'm assuming we're taking a look at some plants, some prairie plants, for instance, that are growing natively. And these prairie plants have a particular locus that I'm calling the A locus that's important in terms of the plants resisting heat and drought. So we're going to say it's the A locus. Now I'm making up this A locus. It's just part of the story, but bear with me. So we have an A locus, and that means that if there are two alleles, big A and little a, there are three types of genotypes among these prairie plants. There's the big A, little a, and little a plants. Now, the next part of the story is, let's assume that climate change is causing the temperatures to raise in the prairies and drought to happen in the prairies. And you're going to have to suspend belief here for a moment, because I'm going to say, instead of it being gradual, it's going to happen overnight. So all of a sudden, what if the prairies became much hotter and much drier? Well, in that case, this A locus becomes important. And these two genotypes are going to survive. But let's say that the homozygous recessive doesn't have the mechanisms that are needed to resist the heat and the lack of moisture, and so they die. Let's just say that's the case. Now let's say that we go out into the field and we take a look at a population of these prairie plants, and it turns out there are 100 plants. And usually, using molecular techniques, we can determine exactly what the genotype is of each of these plants that are outgrowing in the field. And so let's say that for these genotypes, and remember there are 100 plants, 25 of them turn out to be big A, 50 of them are big A, little a, and because there are 100 plants, we figure we can know that there are going to be 25 that are little a, little a. So this is the way the population is structured before climate change happens overnight. Now, let's say that climate change does happen, the situations become dry and hot, and these guys die, and only these survive. Now the question is, what is the next generation of plants going to be like in terms of their genotypes? These are no longer surviving. They aren't going to contribute to the gene pool that's producing the next generation of prairie plants. Only these are. That's surely going to change what the population looks like. So let's see how. So these guys are gone. The way we're going to do this is assume that each of these individuals, of the remaining 75 individuals, is going to contribute 2 gametes to the gene pool. When we take a look at the big A individuals, the only kind of gametes that they can contribute are going to be big A, because that's all they have. So we have 25 individuals, and both of their gametes are going to be big A gametes. So we're going to have in our gene pool, 25 times 2 is 50 big A. The situation is different for the heterozygote. We have 50 individuals, and half of the gametes they produce are going to be big A and half little a. So remember, it's 2 gametes per individual. So of these 50, one of their gametes is going to be big A, the other one is going to be little a. So one big A gamete equals 50 big A, but these individuals are also making a little a gamete, 50 little a. So I need to find a spot up here. What we're going to have in terms of gametes then is 50 plus 50 is 100 big A gametes and 50 little a gametes that go into the next generation. Or 2/3 big A, 1/3 little a. Now we can use this information in order to determine what the genotypes are our next generation. And here's how. We use a Punnett square. And we know that the male gametes, the sperm, 2/3 of them are going to be big A and 1/3 are going to be little a. And the eggs, 2/3 big A, 1/3 little a, same. This Punnett square is a little different than you've seen before because we have frequencies here as well. The reason we didn't see that earlier when we were doing the Mendel Punnett squares is the frequencies of the different gametes of the different alleles were the same. It was all identical, so we didn't need to show the frequencies. Here they're different, and so we need to show them. But now the process is the same. To determine the resulting genotypes, we multiply the frequencies. So for the big A genotype, it's 2/3 times 2/3 is equal to 4/9. For the big A, little a genotype, it's 1/3 times 2/3 is 2/9. And this will also be 2/9 big A, little a. And then the little a genotype will be 1/3 times 1/3 is 1/9. So these are the genotypes that we will see in the next generation. It's going to be 4/9 big A, and 4/9 big A, little a, because we combine these two cells, and then 1/9 little a, that's what's left. So, how do these populations compare? Remember, in the original population where we had 100 plants, we had 25 big A, we had 50 big A, and 25 little a. So this is before. And after, it'll be easier if I assume there's 99 plants in the next generation just because of the way I have to do this multiplication. So now what we have afterwards is 44 plants that are big A, and 44 plants that are big A, little a, and only 11 plants, does that all add up? 88, 99, 11 plants that are little a. So what's happened as a result of one generation of selection? So the very cool thing that I wanted to show you before the camera messed up again is that, remember, we started with a population of 100 that had 25 big A individuals, 50 heterozygotes, and 25 homozygous recessive individuals. And then after this selection against the little a individuals, it really changed things. So that out of a population, this time it was, remember, out of 100, because that was easier to do the division, or out of 99, because it was easier to do divisions by 9. We had, instead of 25 individuals, we had 44. So it really jumped up in terms of the homozygous dominant type. And then the heterozygotes declined a little bit, and the homozygous recessive really tanked, which you'd kind of expect as a result of selection against the homozygous recessive types. But what's interesting is, even though all of the homozygous recessives were wiped out as a result of the heat and the drought, we still had some homozygous recessives the next generation. Because, remember, we've got a homozygous recessive allele hidden in this heterozygote that survives. But when it makes gametes, it's going to make some little a gametes. They're going to find each other and form zygotes and create new little a individuals. So even as the generations continue, we'll continue to have some of these individuals that are no longer fit in the existing situations. So I just wanted to give you a little bit of a look at how population genetics works at just one locus, just as kind of an idea of how it might work when you look at quantitative traits where a whole lot of loci are working at once. Bye for now.